Welcome to Bioinformatics Bytes. In this episode, we'll explore one of the most important file formats in genomics, the VCF file, also called as variant call format. VCFs are everywhere, in research, in medicine, and even in personal genomics like ancestry testing. This VCF file may look like a plain text at first, but inside they hold the story of genetic variation. By the end of this video, you'll understand not just what VCF is, but also how to read one, interpret it, and use it in practice. So let's get going. A VCF file or variant call format file is a special kind of text file used in genomics. It doesn't store the entire genome. That would be way too big. Instead, it records only the differences or variants between an individual's DNA and the reference genome. You can think of it like a spreadsheet. Each row represents a variant and each column tells you specific details. Details like what chromosome it's on, what's the exact position, what's the reference DNA base, what was the alternate base or the change looks like. So instead of handling billions of DNA letters, a VCF file helps scientists focus only on the places where the genome actually changes. Now that makes it much smaller, easier to share, and much more useful for analysis. Alright, so now that we know what VCF file is, the next question is, where do they come from? VCF files are usually created during a process called variant calling, which is part of next generation sequencing or NGS workflow. Here's the big picture. First, the DNA is sequenced and we get the raw reads which is stored in this FASTQ files. Then these reads are aligned to the reference genome, producing alignment files like SAM and BAM files, which we had seen in our previous videos. Finally, specialized software like variant callers, which are like GATK or Freebase or samtools, scan through these alignment to detect differences. The result of this process is a VCF file, which neatly stores all the detected variants. In short, VCF files are the end product of the sequencing and analysis pipeline, and they are the starting point for genetic interpretation. Alright, so now we know what VCF file is and where it comes from. Let's look into the structure of VCF file. VCF file is divided into three main sections. First is meta lines, okay, these ones, which starts with double hash here. These lines describe the background, like which reference genome was used, what software created the file, and the definition of annotations such as read depth or allele frequency. Think of metadata as the instruction manual for your file. The next is the header here, this one in red, okay. So it starts with a single hash. This lines names the column that appears in the data section. Without it, we wouldn't know what each column represents. And finally, we have a data section here. Okay, each row here is a variant, a record of how the sample's DNA differs from the reference DNA. It tells us where the variant is or where the difference is, what the reference and alternate alleles are, how confident are we of this call, like quality scores, and what each sample genotype is. So these are the three sections of our VCF file. Let's talk about each of this section in detail in our next slides. The first part, as mentioned, for the VCF file is the metadata section, and every line begins with two hash symbols. So now let's read all these lines, okay. The first one is file format. In this, it tells us the version of VCF specification being used. In our case, it is version 4. The next one is file date. It tells us when the file was created. Now, dates are often included for traceability in the pipeline. The next one is source. In our case, it is VCF tools. This shows which tool or pipeline produced the file. Some of the common examples are GATK and samtools, which are common sources. The next is reference, okay. So reference here is NCBI 36. Now this line tells us which reference genome was used. Okay, so after this 4 lines, these are some of the other important information. So the first one is info, okay, here you see it's info. So this acts like dictionary, defining what each code means. Now let's look at some examples. Let's say IDAA, okay, which means ancestral allele. Now this basically means the version of the base that existed in common ancestor. It often is determined by comparing across species and is useful for evolutionary studies. Let's not go into the details, but you understand what AA means is ancestral allele. Now let's see what H2 means. H2 means hap map to membership. Now what is what actually it means is the H2 flag means the variant was observed in the HapMap phase 2 project, one of the first large scale studies of human genetic variation. Okay, so I just want you to understand that every line means something here. Even the numbers, you know, when you go into the details, you will understand what number 1 means, what number 0 means, but we are not going to go in that much detail in this presentation. Let's come to the next one, which is format. Okay, now format, again there are certain codes given like GT, GQ, GL, DP. What does all this mean? GT means genotype, GQ means genotype quality Phred scores, which if you remember I had discussed it in a previous videos. GL means likelihood for RR, RA or AA genotypes, and DP means read depth. Now after you understand all this, okay, it becomes easier to read this, right? It becomes easier to read samples. So you can decode the sample columns using your format field. Okay, the next is ALT here. Okay, now this lists the alternate allele or alleles at the position. For example, if it's a deletion or it's a single nucleotide change or there is an insertion, etc. So ALT can also contains symbolic alleles as mentioned, like DEL here, which is deletion. Similarly, it can be DUP to represent larger structural variants. So this in short is what metadata is all about, and it is important to just look into it and understand what each line means. Okay, in our next slide we will go into the header section. So the next section in the VCF file is the header line here. Okay, so I'll quickly go through what each title means. The first one means the chromosome name. It could be chromosome 1, chromosome 2, etc. The second one is the position, the exact base pair position on the chromosome. The third is ID, that is the variant identifier. The fourth is ref, the reference allele at that position in the reference genome. ALT, the alternate allele or alleles observed in the sample. This means the quality score. QUAL means the quality score for the variant call. Filter, which means whether the variant passed quality control filters or failed due to poor quality or depth. Info, annotations about the variants such as depth or allele frequency. Format, the structure of the sample column as we discussed previously. Samples 1, 2, which means the genotype and the related data for each sample. So the header line is critical because without it you wouldn't know what each column in the data rows represent. Alright, so let's take a deeper dive in the data section of the VCF file. Okay, let's take the example of the second row here. Now the first few columns are straightforward, right? Now this shows that the variant is on chromosome 1 at position 2 and it's already known in the database of single nucleotide polymorphism, which is in this case ID RS1. Now let's say if the entry has a dot. Okay, here as you see in the first column row, instead of RS1 or any ID. That means the variant is not in database of single nucleotide polymorphism and it could be novel. Okay. Okay, let's go to the next column, reference, which at this position is C, and the alternate allele, which is T and CT. Okay. The filter says that it's pass, so this variant has met all the quality checks, and that's great. Okay. Now let's go to the next column, which is info. Okay, this is an important one. So the info tells us about ancestral allele, which is T in our case, which I had explained in the previous slide. Okay. Now let's go to the other section, and these are the important sections. Okay. So the format. Okay. Now if you see the format here, it says GT colon GQ, which means genotype colon genotype quality. Okay. Now what does it mean? Let's see first sample. First sample is we see 0 vertical line 1 colon 100. Now 0 refers to the reference allele, and in our case it is C. Okay. And the 1 refers to the first alternate allele, and that is T. So that means that the sample is heterozygous. Notice here you see this vertical line, okay, between 0 and 1, and that is called as phased genotype, which means we actually know which chromosome each allele is on. One chromosome carries the C and the other copy carries the T. So sorry, one chromosome copy carries the C and the other copy carries the T. The number after this is 100, right? And that is the quality score, showing that this call is very reliable. Now let's go to the next example, that is sample 2. And here we see 2/2 colon 70. The 2 points to the second alternate allele, which is CT in our case here. Okay. So both chromosome carry the insertion, making the sample homozygous. Okay. And this time the allele is separated by slash. Now this is unphased phenotype, which means we know both copies of the gene carry CT, but we don't know which came from mom and which came from dad. Okay. The quality score here is 70, which is still decent but not as high as sample 1. So in short, the vertical symbol means phased, so we know the chromosome origin, and the slash means unphased, where we don't know which parent contributed which allele. So that is how you will read the data section. So let's talk about the other part as to why VCF file matters. Now VCF are the universal language of genetic variation. They are used across research, medicine, and even personal genomics. Now let's say in research, VCFs are the backbone of genome-wide association studies, or GWAS. These studies look at thousands of individuals to find variants linked to traits or diseases. Now in medicine, VCFs are used to identify mutations that cause rare disease or influence drug response or drive cancers. Doctors use them to guide diagnosis and treatment. Now in population genetics, VCFs helps us study genetic diversity, ancestry, and human evolution by comparing variants across different populations. So whether you're studying evolution, diagnosing a patient, or exploring your ancestry, chances are the results are stored in VCF file, and that is why it is important to read and interpret, or learn how to read and interpret VCF files. So now that we know how to read a VCF file, let's look at the tools we use to actually work with them. The first one is bcftools. This one is the most popular command line tools for VCF. You can actually view, filter, and merge files. For example, you can use bcftools view to view what's inside the file. You can use command line bcftools filter to keep only the high quality variants. The next one is GATK, which is the Genome Analysis Toolkit. It is widely used for variant calling and filtering. The next one is VEP or ANOVAR, which is Variant Effect Predictor. Okay, this means Variant Effect Predictor. These annotates variants with biological meaning. Let's say if a certain variant changes the protein structure, if there is any change in the regulatory region. So that kind of information you can get through this tool. The next one is one of my favorite, IGV, that is Integrative Genomic Viewer. This is a graphical tool where you can actually visualize reads and variants in their genomic context. The next one is Plink. I haven't used this, but it is commonly used in population genetics and GWAS studies. It can convert VCFs into formats suitable for large-scale statistical analysis. So, in practice, bcftools and GATK helps you manage VCFs, VEP and ANOVAR helps you interpret them, IGV helps you visualize them, and of course Plink helps you with population scale analysis. These are some of the tools that you can use for VCF files. Now before we wrap up, let's talk about a few cautions when working with VCF files. Now VCF are plain text files. Yes, you can open them with Word, Notepad or any text editor, but that's not always a good idea, especially for such big files, because they can be gigabytes in size and may crash your editor. Instead, always use command line tools to open your VCF files. Second thing, VCFs are sensitive to formatting. Every tab, every space matters. If you edit a VCF by hand in Word or Excel, you risk breaking the format. Always use bioinformatic tools like bcftools or vcftools for modification. The next one is compressed and indexed VCF. Often VCFs are distributed as vcf.gz files. These are compressed with bgzip and require an index for fast lookup. Always keep the index file with your VCF. Okay. The next one is sample columns expand quickly. Now in case of multi-sample studies, VCF can have thousands of columns. Be mindful of memory and processing speeds. Finally, use validators. Before using a VCF, run tools like VCF Validator to check if the format is intact. Trust me, this will save hours of debugging later. So these are some of the tips while working with VCF file. Now that's all for today. Thank you so much for listening and please give a thumbs up if you liked the video and comment below. Bye.